Transcription is the process to copy genetic information stored in DNA into RNA. Because DNA is in the nucleus and ribosomes are in the cytoplasm, cells rely on RNA to transfer information, yielding the synthesis of a protein. Shown here is a section of eukaryotic DNA, including a yellow enhancer region, followed by a promoter region, including the Tata box, which is a region made of thymines and adenines located about 26 bases from the encoding region shown in orange. Unlike prokaryotic cells, eukaryotic cells need molecules called transcription factors that aid in RNA polymerase recruitment. The first transcription factor to come in is TF2D, which makes a complex with TBP, Tata binding protein. This then binds to the Tata box. Once bound to the Tata box, two more transcription factors, A and B, get recruited. Together, these form the pre-initiation complex. Once the pre-initiation complex is formed, a mediator complex enters, connecting and stabilizing the promoter region with the downstream enhancer region, bringing with it RNA polymerase and transcription factor 2F. Once RNA polymerase is in place, in the promoter region, TF2H and TF2E are recruited. The mediator complex not only aids in recruitment of RNA polymerase, it also helps with the transition to the open complex, or phosphorylation of RNA polymerase's tail. TF2H allows for the open complex, giving the polymerase access to a single-stranded template. Once the open complex is formed, the mediator complex leaves and the transcription factors disperse. Once the transcription factors disperse, RNA polymerase can escape the promoter region and begin elongation.
A transcription bubble is formed that is 12 to 23 bases long. This allows RNA polymerase to attach to one of the strands and move in the 3' to 5' direction, while generating a complementary strand of mRNA in the 5' to 3' direction, adding bases to the 3' end. Eukaryotic DNA contains tightly packed histones, which present a problem for transcription. To overcome this roadblock, histones must be removed. This is done by the protein FACT, an acronym that stands for Facilitates Chromatin Transcription. FACT removes two chromatin subunits, partially disassembling the histone. The histone is more loosely bound now that some of the nucleosome core is removed. This newly generated space allows RNA polymerase to act and move forward to continue a transcription.
The final step in eukaryotic transcription is termination. The poly-A signal in the RNA itself is recognized by two proteins, CPSF and CTSF, which cleaves RNA to release it from RNA polymerase. On the newly cleaved RNA strand, CTSF leaves, leaving behind CPSF. Poly-A polymerase enters and catalyzes the addition of the poly-A tail, held together by poly-A binding proteins. This poly-A tail is responsible for stabilizing and protecting the new strand of RNA. After the addition of 200 A's to the 3' end of the new RNA strand, which creates the poly-A tail, PAP and CPSF are removed. Capping protein brings in an RNA cap to protect the newly generated RNA strand. The residual RNA is still attached to RNA polymerase. This uncapped end is degraded by the exonuclease. Once in contact with the RNA polymerase, RAT1 dislodges the RNA polymerase, returning it to the closed complex. Now, in its original closed complex, RNA polymerase is ready to be recruited and reactivated again. To summarize, after termination, the products are one new strand of mRNA, parent DNA, and a closed, unphosphorylated RNA polymerase.